Assalamualaikum and a very good day. I'll be to Dr. Amirul Aiman. My name is Anabat binti Aswadi with matrix number 206820. I am from group 3 with 4 other members which are Tasha Elina, Nurul Iza, Nuridatul Aish and Nur Amira Shahira. So today we are going to present about plant response to herbicides. Next slide. Firstly, what is herbicides? Herbicides are a broad class of pesticides that are used to remove nuisance plants that may compromise the growth and the yield of desired crops. Next slide. There are two major categories of herbicides, which are contact herbicides and translocated herbicides. The first one is contact herbicides. It only affects the part of plant that it touches, so it has very limited movement. The absorption through foliage is minimal. Therefore, the application must be made in sufficient quantity to cover the foliage thoroughly. Contact herbicides tend to show symptoms rapidly within 24 hours. While for the translocated herbicides, it moves to the site of action via transport mechanism within the plant, which are xylem and flowworm. The xylem transport water and nutrient from soil, and the flowworm transport the product of photosynthesis. It may take up to 2 weeks for the symptoms to develop depending on the herbicide rate, condition, and the plant species. So that's all for me. I will pass to our next presenter. Thank you.
So I will continue with the plant's response to herbicides in terms of its morphology. Firstly, herbicides are root growth inhibitors. How did that happen? These herbicides inhibit cell division as part of their mode of action, which ultimately blocks root extension and growth. Their site of action is the microtubule and is marked by the assembly of the herbicide tubulin complex inside the microtubules. This complex inhibits the polymerization of microtubules during assembly but remains unaffected during depolymerization. A loss in the structure and function of the microtubule causes cell death by compromising the cell wall formation due to the non-alignment of the spindle fibers and non-separation of the chromosome during mitotic cell division. The benzamide, benzoic acid, dinitroaniline, phosphoramidate, and pyridine act by causing the loss of microtubule formation, leading to the obstruction of cell division and elongation, as evidenced by swelling of the root tips. Next, herbicides are also shoot growth inhibitors. Next slide. The herbicides are applied as part of the soil preparation and act effectively before the grass and broadleaf weeds emerge. The site of action of the herbicide is at the location of the lipid synthesized machinery in the cell membrane. These herbicides are represented by the chemical family of phosphodithioates and thiocarbamates and inhibits the biosynthesis of lipid, fatty acid, proteins, isoprenoids, flavonoids, and gibberellins. The site of action of the chemical family of fluoroacetamide, acetamide, oxyacetamide, and tetrazolinone is at the very long fatty acid located in the cell membrane. This herbicide conjugate with acetyl-CoA and certain sulfhydryl-containing molecules via thiocarbamate sulfoxide, which inhibit the long-chain fatty acid during the seedling shoot growth stage of the plant and affect the weed's pre-emergence. That's all for me, and I will pass to the next presenter.
Thank you, Tasha. And I will talk about the physiology in the plant response to herbicide. Herbicides are taken into the plant through the leaves and through the roots. Some herbicide can be absorbed only through the leaves and some can be absorbed through the roots. Other can be absorbed both ways. Uptake into one or other organs, but not both, illustrate how the physiological process absorption can be selective. Herbicides that are by the roots are applied to the soil. In practice, we refer to these types of chemical as soil applied herbicide. So how does root absorption occur? Chemicals are taken into plant by both passive and active uptake. Passive uptake occurs by diffusion from high to low concentration and is not under close metabolic regulation by the plant. Active uptake requires an expenditure of energy by the plant and is metabolically regulated. Chemicals, including plant nutrients, are absorbed into the roots by both active and passive uptake. It is generally concluded that a herbicide enters the roots in the same way as plant nutrient. If a mechanism is functioning for the uptake of nutrients, then the plant will not be able to restrict an herbicide that is taken up in the nutrient stream. Next slide. Okay. This is the example of the chemical composition and structure of the cuticle may vary between plant species and this can account for differences in herbicidal penetration and activity. The left side, thick cuticle prevent absorption while the right side, thin cuticles permit good absorption of herbicide. Next. Okay, next is a translocation. Movement of a chemical within the plant is called translocation. Herbicide may have no activity at the point of uptake but may be very potent inhibitors in other parts of the plant. Therefore, many chemicals must move once they enter the plant in order to exhibit their effect. Other herbicides do not move at all and still exhibit herbicide activity. Such non-mobile herbicides are called contact herbicide because they are potent at the site of uptake. Both foliar and root apply herbicide may be contact herbicide. Next is the component that involved in translocation process. A discussion of the structure and function of plant conducting tissue is necessary. Both living and non-living systems are found in the plant and each support a different process of translocation. The non-living part of the plant is called apoplast. The cell wall, fiber, air space, water, and the xylem, a conducting tissue, are part of the apoplast. The living part of the plant is called the symplast. The symplast include the cells that and their components including the flower, also a conducting tissue. Next slide. Okay, the next is a site of action. Herbicide activity may occur at many levels in the plant. Some herbicide reactions are very general and some are quite specific. General types of reaction are those which interfere with whole system, for example, the dissolution of a plant, critical waves, or the destruction of membranes. On the other hand, herbicide may have a very specific activity at the cellular or subcellular level in the plant. Although it is not necessary to know the details of how herbicide inhibit specific reaction in order to use them in the field. Such knowledge is extremely useful in the development of new product. In addition, knowledge of specific reaction enables us to better understand why chemical work under one set of condition and not another. That's all from me. I will pass to the next presenter.
Thank you, Aish. So I will continue about the effect herbicide plant. First is herbicide can injure foliage, shoots, flowers, and fruits. If injury is severe enough, either from one incident or repeated exposure, it may reduce yield, produce fruit quality, destroy ornamental on the sweet plant, and occasionally cause plant deaths. And herbicide symptoms may be visible for a few days to several years depending on the herbicide involved, plant species, stage, and rate of growth, environmental and soil condition, and cultural practices. As we can see the picture at bottom, the picture shows the leaves tend to stay flat but have enlarged vein and causing distortion of new growth on perennial. Next slide. Next, herbicide can act by inhibiting cell division, photosynthesis, or amino acid production. For cell division, there is cell division inhibitors, which is herbicide are applied at the soil and carried to the leaf by transpiration. For example, atrazine, if the use of atrazine exceeds the prescribed dosage, then it will inhibit the process of photosynthesis. The symptom is injured leaf turn yellow or white, then often translucent. Second is cell division inhibitors, whereas metolachlor, for example, these herbicides are applied to the soil to control target vegetation before emergence by inhibiting root growth. If exceed, then rapidly turn yellow or pale and may look water salt. Then they dry up. Lastly, herbicides like glyphosate, where these herbicides are applied to the foliage and transported with sugar to metabolic site where they inhibit amino acid production. If the herbicide exceeds, the plant treated with glyphosate turn to yellow in 5 to 7 days, then turn to brown and die in 10 to 14 days. So that's all for me. I will pass to the next presenter.
Um, okay, Amira. So for the conclusion, herbicides are phytotoxic to crops, especially when the concentration used is above the recommended level. The selective application of herbicide can cause biochemical and physiological damages, and it can damages that can cause oxidative stress and can counter damage other processes. I think that's all from us. Thank you.